Okay, I'm sorry. I apologize. Something dreadful happened to the recording. So this is going to be divided into 2 recordings, and you'll have to excuse the swearing and cursing at the end of the last recording when I realized that something had gone wrong. In any case, we recommence where we left off. So we saw, first of all, an important thing about RNA: it can act as an enzyme, and one of its functions is that it can act as a replicase. It can act as an enzyme which will replicate itself. And that is, of course, the critically important step. Any genetic information which was contained in those early those early nucleic acids, any information contained in it could be handed on as a copy to succeeding generations. That was a critical step. Evolution of DNA was a later development. Again, we don't understand all of these steps. We just know that they have to have taken place, and we do have experimental evidence that some of our ideas are correct. So much of our understanding about the evolution, especially about macroevolutionary processes, comes from the fact that they are recorded in the fossil record. And the fossil record, there are many different kinds of fossils, but the majority of the fossils that we rely on are fossils which are found in sedimentary rock. And the fossil record is based on us examining those sedimentary deposits and realizing that organisms which are deposited early on are further down in a sedimentary record than those which are deposited later and which are higher in the sedimentary record. There are other kinds of fossils as well, which are also important and which can often provide us with really important information. This is the most common kind of fossil, and these are impression fossils, such as plant fossils. When we look at them, what we're seeing is almost like a photograph. The organism often has been entirely carbonized. There's nothing left of the original molecules, but an impression is left in the rock, sufficient for us to be able to examine it and identify morphology and everything. That very, very fine details of morphology may actually be preserved. There is another important set of fossils, and these are fossils where the original organic material, which would have, under normal circumstances, would have decayed, has instead been preserved and has slowly over time been replaced, often with silica compounds, which we refer to that as petrification, turning to rock. An example would be things like petrified trees, which you find in places like Arizona. But this also occurs with animal fossils, and we find these petrified, silicated fossils are very, very important, and again, very often preserve very fine detail of morphology. There are other kinds of fossils which we refer to as trace fossils, not fossils of the organisms themselves, but signs that they were there, such as, for example, very commonly, footprints and also burrows, that sort of thing. Very rarely, and only under very special circumstances, organisms may be frozen. Now, this in our time, this is becoming increasingly important, because many of these fossils are frozen into permafrost in the high latitudes particularly of Europe. And they are now, due to global warming, those permafrost deposits are beginning to thaw, and more frequently we are finding things like very well preserved organisms such as wolves, mastodons, early elephants, and all of these kinds of organisms. The last one is one which you probably know from the movies, most of all, from Jurassic Park. This is organisms which are preserved in amber. Amber is tree gum. It is a gum which is produced from trees. Most of our amber actually comes from coniferous trees, and the amber becomes fossilized over time. But when it drips down trees, it's very attractive to insects. And insects were very, very frequently caught in it and then themselves fossilized. Both of these, the frozen fossils and these, the amber fossils, have a potential importance that they may have DNA which we could extract. And that can be very, very important. And in fact, this has been done. The DNA molecules themselves, unfortunately, very rapid soon after death, unless the organism is snap frozen, which doesn't usually occur. It usually takes some time for them to freeze. But DNA, unfortunately, breaks down relatively quickly. Nonetheless, there may be fragments of DNA and often mitochondrial DNA, which can be analyzed. So these are very important fossils. This is a wolf pup, poor little thing, but you can see perfectly preserved, perfectly recognizable. And they have to be very carefully treated because once they start thawing out, they begin decaying very quickly. Okay, so the fossil record allows us to see these huge changes that have taken place over time. We notice several things. First of all, most of the very old organisms are nothing like anything that is present today. And some very, very major dominant groups, the trilobites, for example, the ammonites, the dinosaurs, these groups which were incredibly important, very numerous, dominant in many, many different ecological niches, have entirely disappeared. And it is a lesson to us that even the highly successful groups may be presented with evolutionary challenges which they cannot that most of the group cannot surmount. But usually, in those groups which are struck with a mass extinction like that, there are some which do survive. They act as ancestors for succeeding groups, which then rapidly develop from those surviving ancestors. Unfortunately, the fossil record is very, very incomplete. There are huge gaps in the fossil record. Fossilization is extremely rare compared to how numerous organisms actually are. Very few of them get fossilized. Second of all, even if fossils are formed, the Earth is continually changing. There's continual geologic change, and many of the fossils end up just being destroyed. Thirdly, fossils are often difficult to find. They are buried deep in very hard sediments. We only have sporadic and cursory access to those sediments. And so we only find a small proportion of the fossils which are actually available, which are actually present. So as the fossil record, fascinating though it is, we always have to realize that it's very incomplete and that we need any conclusions that we draw from the fossil record, we need to caveat. We always need to say, well, the story could change if we found other fossils, but this is what the evidence appears to show for us at the moment. So the other thing is that the fossil record is strongly biased. Okay, it's biased in favor of those organisms which, I'm sorry, I really seem to be struggling with this technology. The fossil record is very biased. Those organisms which have existed unchanged over a very long period of time, those ones statistically stand a better chance of having been fossilized. Second of all, the fossil record tends to pick out those which are very abundant and which are widespread. It's seldom, the chances of a rare organism being fossilized and us finding it is very remote. Thirdly, is those organisms which have hard parts to them, a skeleton or an exoskeleton, something like that, those tend to be fossilized much better than the soft parts. Soft parts rot away very, very quickly. And unless an organism is preserved under very special circumstances, for example, if an organism, a soft-bodied organism, happens to fall into a completely anoxic sediment, a sediment with no oxygen in it, it tends to stand a better chance of being preserved and then entering the fossilization process. Under normal circumstances, soft parts rot away very quickly. Hard parts persist and are much more liable to be fossilized. When we look at the whole chain of all the fossils that are available to us, even very early on in the 19th century, it was realized that when we look at the fossil record from very distant times up to more recent times, we see that there are giant changes, big jumps in the fossil record, where major changes take place in the groups that are present. And these served as dividing points. It allowed the early geologists, paleontologists, to classify sediments by what they saw present, and also to begin to associate some sort of date to them. In the early days, all they could do is say, this is very, very much older than that. But as science has proceeded, we've actually been able to assign dates to, to these different layers. So first of all, we divide that geological record into eons. Here they are here. They are the Hadean, Archaean, Proterozoic, and Phanerozoic. Phanerozoic being the most recent. That is us. We are still in the Phanerozoic eon. And Hadean is that period of time when no life existed, or life maybe existed in proto-cell form or something like that. We do not have a fossil record in the Hadean. In the Archaean, we begin to see fossils forming, as I'll show you in a minute. And at the end of the Archaean, by the end of the Archaean, we begin to see true cells forming. And we go into then the Proterozoic. This is the era of, or the eon of early life. The Proterozoic is early life. And we'd have fossils present, mostly prokaryotes from the Proterozoic. The Paleozoic is the era of ancient life. Now, this is the era. The Phanerozoic is the era where we see our recognizable groups. These are the groups which we see, many of them present still to this day. This is the era where we see the phyla and everything, which we still see today. So the eons are divided into eras, and these eras here, the boundaries are actually largely determined by major changes in the groups of organisms that are present. The eras are divided into periods, and this era here, the Cenozoic, divided into these three periods. These are also divided into epochs. These are the most recent. These are the most recent divisions of time. And actually, if we look through that, we find that in fact, many of those boundaries between the different time periods, very often these were the result of mass extinctions. We see very rapid die-off of particular groups, even if they were majorly dominant. And we see then subsequent to that, new groups arising. Let me see if I can show you a couple. Here, for example, there was a very early type of fauna called the Ediacaran, which died out completely at this boundary here, which led into the Cambrian. So we moved from, this is Proterozoic, we moved from Ediacaran fauna, then into the Cambrian. And we believe that one of the contributing factors here was the development of predation. I'll explain that a little bit more in a while. The Paleozoic ended here. It ended with a mass extinction. We do not understand the mass extinction exactly. Some people think it was just due to worldwide geological activity and sudden volcanic activity, but a very important group, some very, very important groups died out here. This is the Permian extinction. And at this point here, a huge group of reptiles called the mammal-like reptiles, which had been very, very dominant and very important, died out entirely. And what was left behind were our ancestors, the early ancestors of the mammals. They didn't succeed because the niches that had been filled by the mammal-like reptiles were filled by the dinosaurs. And the dinosaurs were wiped out here, at this mass extinction here. And their loss led to this small group, the mammals, exploding into all of the ecological niches here in the more recent times of the Cenozoic. So that's just an example of how, if we look through this fossil, if we look through all of the fossils in this fossil record, we would see these changes, okay? We see massive changes here. Here, this point here, we see two huge groups disappear, the mammal-like reptiles, and then in the marine systems, many, many marine groups disappear, but the one which disappears entirely is a group of arthropods called the trilobites, hugely important, very, very common throughout the seas. All of a sudden, they all die out here. Here in the Cretaceous, of course, you know, we see the die-off of the dinosaurs, but we see subsequent to that, the development of the mammals. Along with the dinosaurs, here at this point here, we also see disappearing major marine group called the ammonites, which were cephalopods. They're related to the octopus, squid, etc. So the lesson of this geological record is extinction is actually the rule, but extinction always acts as the prelude to major new developments of living forms. So it's very common to refer this to this time. It's inconceivable, the length of time that this is, okay? 4.6 billion years since the formation of the Earth. And for all our modern forms of life, we trace from about 550 million years ago. So these are huge time periods that we are talking about. It's very difficult to visualize how that is. So we often refer to some analogous system, a clock, something like that. And here, for example, if we look at, we just think of about a 12-hour clock, then about a 60 minutes clock, and we go backwards and say, okay, here's 12 o'clock. That's our present time. Humans, 11:59 and 30 seconds, maybe, we first formed a species. We are a very, very, very recent addition to the history of life on Earth. If we look, in fact, colonization of land, widespread colonization of land by multicellular eukaryotes took place, let's say, what would we say, about 5 minutes ago. The formation of animals, the development of animals themselves, maybe 10 minutes ago, 9 minutes ago, something like that. And in fact, the development of multicellular eukaryote forms only 15 minutes ago. Look at the length of time of the Earth's history where there were no multicellular eukaryotes. It's by far the bulk of the Earth's history. There were no multicellular eukaryotes of any sort. And in fact, eukaryotes themselves only formed on our 60-minute clock 25 minutes ago. When we look at the history of the Earth, in fact, the prokaryotes have existed for a very, very long time. They actually came to be the first prokaryote cells quite rapidly compared to everything else. And they would have been formed, well, within 15 minutes on this clock, within 15 minutes of formation of the Earth, 45 minutes ago, if we're going around this way. So that is the prokaryote. We're going to hear about these changes in more detail in a minute. But this gives you an idea. In actual fact, the Earth is very old, but many of the forms that we think of as being so common and present on the Earth's surface have not been there for that long. Okay, they are in fact relatively recent. So here are fossils of the first true cells. These are prokaryotes. These are bacterial cells of one sort or another. And we have no idea really about the internal structure of these cells, but they certainly, from the outside, they certainly are recognizable cells with a clear cell membrane boundary and maybe even a cell wall. We're not exactly sure. But they certainly look prokaryote. There are many prokaryotes still around which closely resemble these. These are some very, very ancient rocks from Australia and from Africa, about 3.9 billion years old. So though these cells here are heterotrophic, they are strictly heterotrophic. They were surviving by utilizing the molecules which were present, which had been created abiotically in that primordial soup. They were busy just taking those in and utilizing them in their metabolism for growth and everything else. There was a major problem, and that was that resource was very limited. These living forms were extremely successful. They must have been very common because we find them widely in these very ancient rocks. But the problem was that they were utilizing their own food source and they were not replacing it. Instead, they were taking in these complex molecules which had been made by abiotic processes. And very often they were breaking them down during their metabolism. They were creating simple compounds which were released as waste. But there was no new source of material, and above all, there was no new fixation of energy. They very soon began to run out of their food resources. And that evolutionary pressure was so strong that, in fact, this group began to arise quite soon after the Earth's formation. We find them 3.5 to 3.7 billion years ago. We recognize in the fossil deposits these organisms. which are photosynthetic prokaryotes. They are prokaryotes which could take simple compounds, carbon dioxide and water, and they could use the sun's energy to form organic compounds, which they could then use both to build them in anabolic processes to build and grow, but they could also use in catabolic processes to derive energy. And these early organisms, these were the early cyanobacteria, the photosynthetic bacteria that we still see very, very commonly. These formed a good fossil record for one reason, and that was they tended to, when they divided, grew and divided, they tended to remain associated with one another. And we see exactly the same thing taking place under modern circumstances. Have a look here. This is an area in Australia called Shark Bay, very famous biological site. And these, what look like rocks here, are in fact huge colonies of these cyanobacteria. Only the very surface is living. The bacteria actually can move, and they actually migrate to the moist, lit areas at the surface. When they're there, they make a capsule around themselves, for example, which is protective. But that capsule also keeps them together, and the capsule tends to trap sediment, it tends to trap sand and that sort of thing. When they start to get shaded out, they move out to the surface again. And this continual movement, growth, movement, growth, movement, growth, movement, growth, movement. When we cut through, we see that they form layers like this, which accrete to one another. And that is very, very slowly. And that is what produces this growth. A stromatolite this size is probably thousands of years old. But they fossilize very easily because they're already more or less rock. They're more or less cemented sediment already. It's only the very surface which is living prokaryotes. So they're fossilized very, very well. And fossil stromatolites are recorded in many parts of the world, Canada, Africa, Australia, and they sometimes are extremely old, 3.5 to 3.7 billion years. So we know that these were operating as far back as that. So that's fine. These photosynthetic prokaryotes, they're doing something critically important. They are fixing energy from the sun into biological energy. They're taking simple compounds, they're putting them into complex compounds which other things can utilize. But there's a huge problem associated with this process. And that is that this process, which you recognize is photosynthesis, begins to release oxygen. And oxygen is phenomenally dangerous to biological molecules. It is very, very reactive. And this led, in fact, to the first mass extinction. And the first mass extinction was caused by the presence of free oxygen, O2, which began to accumulate in the seawater. And that free oxygen killed most of the prokaryotes. And the prokaryotes that survived the presence of that free oxygen were those which could either retreat to little habitats where the oxygen was excluded, like the sediments, for example, or more importantly, those which were able to develop a system to deal with the oxygen. And this pressed them, the evolutionary pressure was to those organisms which could satisfactorily deal with the presence of oxygen in their environment. Now, that first burst of oxygen very quickly got sopped up by the free metal ions and things which are present, like iron in seawater. And we see this in the geological record. We see the massive accumulation of iron oxides, for example, very rapidly as these photosynthetic prokaryotes begin to release oxygen. But eventually, the oxygen itself began to accumulate and increase in concentration. And once the water was saturated with oxygen, then the oxygen began to accumulate in the atmosphere. And the atmosphere now changed from that early reducing kind of atmosphere to one which became richer and richer and richer in oxygen. And that increase in oxygen in the atmosphere is recorded to be about 2.7 to 2.4 billion years ago. So here we are here. Atmospheric oxygen is beginning to accumulate. Notice something, and that is that this atmospheric oxygen, there is nothing up on land. There are no living things up on land yet. Everything is aquatic still. So once terrestrial evolution began, it did so in an oxygen-rich atmosphere. So this is called the oxygen revolution. This sudden accumulation of oxygen is called the oxygen revolution. And we look through the geological record, you can see it quite clearly. This period here is when this massive deposition of iron oxides, which we mine now as iron ore. This is when they were all laid down. So here it is here, about 3.7, 3.8 billion years ago here. Subsequent to that, there have been various times when oxygen varied somewhat, but this was the first major accumulation of oxygen in the atmosphere. We see other changes once evolution on land takes place. And plants start to develop photosynthesis, and we'll see another burst, but this is the major burst here. And remember, this oxygen revolution led to a mass extinction. It's just that the mass extinction was of the only living things, which were the prokaryotes at that time. Now, I said we had survivors of the prokaryotes. And those survivors either retreated to anaerobic environments where there was no oxygen present. And those survive nowadays as the anaerobic bacteria. They're still a very important group of bacteria. The other group that survived were those which had the evolutionary potential to develop ways of dealing with the presence of oxygen. And the most important strategy of all to do that was the development of cellular respiration. Cast your mind back and remember that cellular respiration is the utilization of oxygen as a terminal electron acceptor in a chain of events which leads to the formation of ATP, adenosine triphosphate, the useful biological source of energy for most metabolic processes. Oxygen there is being utilized as a terminal electron acceptor. And what is it being turned into? It's being turned into water. It is being reduced. The oxygen is reduced to water, H2O. And that is harmless. The oxygen is extremely harmful. It has the potential to be extremely harmful. H2O is innocuous. So the cellular respiration developed initially as a strategy to remove oxygen and turn it into water. But along the way, there, over evolutionary time, was the possibility of modifying this process to actually produce useful biological energy. And that's exactly what happened. So the first eukaryote fossils that we see are of single-celled organisms. And in fact, the eukaryote cells, single-cell eukaryote cells were dominant for a very, very long time indeed in the Earth's history. They first developed about 1.8 billion years ago. And we remember back the difference between the prokaryotes and the eukaryotes. Both have a cell membrane. But in the prokaryotes, the cell membrane is the only membrane. The cell membrane for both of them is a boundary between the inside of the cell and the outside. It allows conditions inside the cell to be different to outside of the cell. But in the eukaryotes, there are other membranes, and they enclose organelles within the cytoplasm of the cell. But the function of those membranes is much the same. It allows the conditions within the organelle to be different to the conditions outside in the cell membrane, in the cytoplasm. This allows these organelles to specialize, and it allows them to specialize their function. But when we look at the organelles which we have present in eukaryote cells today, the most dominant one is, of course, the nucleus, which contains the bulk of the nucleic acids of the cell, and is the site of RNA synthesis from DNA, et cetera, the nucleus is also the location where DNA is replicated during cell division. But look at the other organelles, and we see something strange. Look at two in particular, and those are the mitochondria, which are present in plants and in animals, and the chloroplasts, which are present only in plants. And when we examine those organelles, we realize something really important. They have double membrane around them. That's the first thing. The second thing is the inner membrane is slightly different to the outer membrane. The inner membrane is more like a prokaryote membrane. There are some differences between eukaryote and prokaryote membranes. When we look inside the cytoplasm of chloroplasts and the mitochondria, we also see that they have their own ribozymes. Not only that, they have their own DNA. Their DNA is in the form of a chromosome, and the chromosome is circular. And the chromosome looks very much like a prokaryote chromosome. So do their ribozymes. In modern times, chloroplasts and mitochondria can't exist independently of the cell. They are unable to produce all of the things that they require. They can produce some of their own proteins, but not all of them. All of this leads us to believe that those, in particular, chloroplasts, mitochondria, and other in plants, some other organelles which resemble the chloroplasts for one reason or another, we believe that these were all acquired by endosymbiosis, that mitochondria, chloroplasts were once free living, and that an early eukaryote cell acquired these, incorporated them into the cytoplasm, and developed a mutual symbiosis, which became so profound that now it characterizes the eukaryote cell. So the original chloroplasts, the original mitochondria, these were endosymbionts. Now they cannot exist outside at all. The partnership now is irrevocable, and it's characteristic of the eukaryotes. Here are some very, very early eukaryotes. I qualify that. These are some of the earliest eukaryotes we find in the fossil record. They're probably not the earliest eukaryotes at all. They're probably quite well evolved eukaryotes. Nonetheless, we recognize that they not only have, they probably had cell walls which allowed them to be fossilized easily. They have strange forms. We even see some which look like they are almost beginning to be colonial, cells associated with one another. So these about I believe these fossils are about 1.5 billion years old. Okay, here's just a summary of of why we believe chloroplasts and mitochondria are endosymbionts. Those eukaryotes were extremely successful. Those early eukaryotes, which were single-celled, were extremely successful. They became abundant very, very quickly. But the next step in evolutionary, in macroevolution, was when these cells began to associate with one another to form multicellular forms. And from those multicellular forms ultimately developed all of the groups which we see today, the algae, the plants, fungi, animals, etc. So this development of multicellularity is an extremely important step. The earliest multicellular organisms are a group of photosynthetic organisms which resemble what we believe were red algae. And that's about 1.2 billion years ago. By about 600 million years ago, we do see the roots of the modern groups which we see today. But intervening, there is, in that Proterozoic eon rather, there is a lengthy period of time when a very strange fauna existed. And this is what we refer to as the Ediacarans. So let's just have a look at our clock and see where we are. We've seen the development of the single-celled eukaryotes. Here, if we're thinking of 60 minutes, that would have been about 25 minutes ago. Now we're having a look at the development of multicellular eukaryotes. Look at the length of time that the eukaryotes were predominantly single-celled, a very long period, almost half of the time that eukaryotes have existed. In fact, they were single-celled. But here are the multicellular eukaryotes, about 15 minutes ago on our 60-minute clock. So relatively recent. And here we see the two early forms of these multicellular. This is a red alga here, a filamentous red alga. Not much different to the filamentous red algae that we still see around today. This, on the other hand, is one of the Ediacaran animals. And the animal form, now, let's just go back here. Here are the animals here. So we're talking here about 700, 800 million years ago. They developed this very strange fauna. We don't exactly understand how they functioned. The current thinking is that they were floating forms. They may have been associated with photosynthetic organisms in a symbiosis. We're not exactly sure, but they don't appear to have any way of eating. So they were either absorbing molecules from the outside, or else they had some sort of symbiosis, which allowed them to exist. They're mostly these flat forms with no really distinct morphology that would tell us exactly how they functioned. This is the Ediacaran fauna, but there were many associated things. But throughout this whole period, there's not a huge amount of diversity either. These fossils are found in Africa and in Australia as well. I'm not sure about Canada, maybe from Canada as well. But have a look here. Here's when the Ediacaran, this is right here, the Ediacaran fauna. Very, very suddenly, towards the end of the Ediacaran, this Ediacaran period, a set of groups begin to appear in the fossil record with radically different morphologies. And the Ediacaran fauna, this flat floating fauna, very suddenly disappears. And here, at this boundary here, all of a sudden there is an explosion, just a massive adaptive radiation. We just see incredibly diverse forms suddenly arise in a very short period of time. So something major happened here, which wiped out the Ediacarans and led to this sudden appearance of many, many different forms. Now remember, the roots of that diversity lie at the end of the Ediacaran. It starts to appear at first, gradually, the roots of these groups, but suddenly at this point here where we move into the Cambrian period, all of a sudden it explodes in diversity and in numbers as well. Just the most bizarre, strange forms appear. But when we examine them, we recognize the origins of many of our modern lines, especially of the animals and especially of the arthropods. We recognize that their roots lie there, the end of the Ediacaran, and then in this burst of diversity in the Cambrian. It's so marked that it's referred to as the Cambrian Explosion. And if you go to Wikipedia, even if you want, go in and find some reference to the Cambrian Explosion and have a look at some of the really, really bizarre and wonderful animals which suddenly came to be. Here's one. This is called hallucinogenia. Literally because it looks like a hallucination. Here are fossils of hallucinogenia here, and here is a reconstruction. Many of these fossils from the Cambrian, it's very, very difficult to say exactly what they are, exactly which group they belong to. But in many of these very bizarre groups, we recognize some of the morphologies that are present in our modern day forms. The Cambrian explosion is around about here. And here are the groups which become established, the end of the Ediacaran and then into the Cambrian. Sponges, those are a very, very old group and believed to be actually ancestral to most of the animal groups. The ultimate animal ancestor may have been something that resembled a sponge. Towards the end of the Ediacaran, all of these groups began to appear. The Cnidarians, jellyfish, sea anemones, all of those. This group, which is our group, the chordates, closely related to the echinoderms, the starfish and that sort of thing. Many, many groups of mollusks, and very, very importantly, the arthropods. And as I say, if we were to look, we'd see that each of these groups just exploded here into huge diversity. They may have existed towards the end of the Ediacaran, but they developed huge diversity as the Cambrian began. What was it that actually drove this sudden strange explosion of diversity? Well, we think, in fact, that it was something which to us may sound relatively simple. And that was the ability to catch and eat somebody else. Instead of just depending upon the environment as your source of food, either absorbing molecules from the environment, or else maybe having a symbiosis with something which fixed the sun's energy in photosynthesis. Instead, there was the development of the ability to actually catch a neighbor and eat it. True heterotrophy. To actually actively catch, eat, and digest molecules which had been made by another organism. This is predation. And we believe that the development of predation towards the end of the Ediacaran period that actually led to that Cambrian explosion. There's very little evidence prior to that of any predation, of any mechanism of predation. But when we, towards the end of the Ediacaran, and especially in the Cambrian explosion, we suddenly see the development of major ways of catching other organisms and eating. We'll see the development of fearsome jaws, fearsome feelers, and things like that, which could catch and grasp things while you ate them. But likewise, if the evolutionary pressure is driving the development of mechanisms of catching things and eating them, likewise, evolutionary pressure is going to be driving organisms to develop mechanisms to evade predation, to keep predators off. That may explain, for example, all of these spines and things, and organisms like hallucinogenia. These are developed to try and fend off predation from other organisms. So we really do believe that the evolution of predation was a major driving force for this explosion of diversity. The next important step to take place was the movement of organisms up onto land. And this took place about 3.2 billion years ago. In fact, that initial that's a long time ago, 3.2 billion years ago. But for a very, very, very long time, prokaryotes were the only ones which were present on land. They could survive close to water. They could maybe even survive a bit further in. But the only living things up on land were prokaryotes. And that remained the case until about 500 million years ago. You realize 500 million years ago, it sounds to us like an enormously long time ago. It's relatively recent in geological terms. And here it is here. And the first organisms to move up onto land were in fact the plants. And we'll hear about that later on in the semester when we talk about plant evolution. They were simple plants which moved up onto land, but their movement onto land was tremendously important, tremendously significant, because they caused conditions on land to change so radically. They began to contribute oxygen to the atmosphere. They began to change the margins of water bodies, and they enabled the penetration ultimately of animals onto land as well. The movement onto land was not a simple thing. In water, you do not have to fight gravity. Water supports the body. You're effectively weightless in water. As soon as you move up into land, you have to develop strategies for resisting gravity. A jellyfish in the ocean is a beautiful thing. It is perfectly functional. It has a very elaborate structure, which is able to maintain simply because it is effectively weightless. Move that jellyfish onto land, suddenly gravity compresses the jellyfish into a slab of useless jelly, of useless tissue which cannot function. To move onto land, you had to have the evolutionary potential to develop structures which would allow you to resist gravity. You also would have to resist dehydration. You would also have to adapt your reproduction in some way. And we see the start of all of these kinds of adaptations, especially things like dehydration, that sort of thing. We see them occurring virtually as soon as things moved up onto land. We begin to see those kinds of adaptations. Okay, I'm going to stop there for today. I know I'm going to have to spend some more time in the next class trying to complete this, but this is a very important section to set the tone for all of the material that we're going to be reading. Again, I urge you to please go to your textbook and read. I'm just literally skimming through these issues. I am going to set one extra credit exercise for you for this section, and that is I have not discussed at all how we date those sediments and how we date fossils. And I'm going to ask you to do that on your own. There's an explanation in your textbook, but you can gather other information from outside sources as well. I will post that early next week for you, okay? But that will be your first extra credit assignment. And with that, I wish you a happy weekend. Please remember, wear your mask, socially distance, keep it up. Make sure that you stay as safe as you possibly can. The vaccine is coming, but we have got a long time to go before that vaccine hits our arms. And we need to keep ourselves and everyone around us safe. Okay? I wish you all well.